NBIA, Natural Bioscience Intelligence Agency. The science is real. The mission is yours.
22 minutes past midnight. Lincoln, Nebraska. Agricultural Research Services Food Safety Lab. I'm standing over a chromatography output that the FDA has never seen. The sample is a frozen potato side dish. Oven-baked line, 41 states, 6,000 retail locations. The acrylamide concentration in this batch is not a trace contaminant. It is a function of chemistry, predictable, reproducible, and completely legal to sell.
This isn't one brand, Viper. Acrylamide forms in every product category where starch and amino acids meet high heat. Potato chips, breakfast cereals, commercial crackers, roasted coffee, bread crust. The convenience meal is the highest volume delivery system, but the chemistry is the same across all of them.
Understood. So we're not looking at a rogue product, we're looking at a rogue decision to not fix it when they had the method to do so.
Biochemistry is indifferent to business cases, Viper. The mitochondria don't care what the label says. The solution is also real and specific. You'll have it before this episode ends. 18 hours earlier, Geneva.
The European Prospective Investigation into Cancer and Nutrition. 500,000 subjects. The largest nutrition biomonitoring study ever assembled. And buried inside it, a variable nobody thought to extract. Acrylamide hemoglobin adduct concentrations. Self-reported fatigue severity scores. Same cohort. Same decade. Never overlaid. Until now. The subjects carrying the highest acrylamide adduct burden report the most persistent, treatment-resistant fatigue. The dietary pattern predicting the highest adduct load, convenience food, daily, across all 3 meals, working adults, 35 to 65. Too busy to cook. Too tired to wonder why they're tired. Joining mobile briefing. Lyon to Lincoln. Secure link established.
Acrylamide. Full name, 2-propenamide. Not a synthetic additive, a chemical consequence. Frost mechanism.
Before the molecular detail, think of your mitochondria as a turbine. That turbine runs on electrons passed along a chain of protein complexes. Complex 1 is the entry gate. Acrylamide's metabolite is a wrench thrown into that gate. Here's exactly how it gets there. Acrylamide forms when asparagine reacts with reducing sugars above 120 degrees Celsius during baking, frying, and roasting. The Maillard reaction, the same chemistry that browns bread and crisps potatoes. CYP2E1 in the liver converts acrylamide to glycidamide, a reactive epoxide. Glycidamide forms covalent adducts with cysteine residues on NADH dehydrogenase subunits inside mitochondrial complex 1. Adducted subunits cannot transfer electrons efficiently. Electron leak increases. Superoxide radicals generate. ATP output falls.
So the same reaction that makes the food taste good is what damages the mitochondria?
Correct. The browning and the biochemical harm share the same chemical origin. One is visible, one accumulates silently over years.
The IARC EPIC data shows this accumulation is not theoretical. The adduct burden in high-convenience food consumers is measurably elevated, and it tracks against fatigue scores with a consistency that does not require statistical generosity to read.
The formulator, Frost.
A frozen convenience meal brand, 4th largest by US retail volume, 22 SKUs, 41 states. The internal food science team proposed asparaginase enzyme pre-treatment in 2018, a commercially available method that reduces acrylamide formation by up to 90%. Cost, 4 cents per unit. Retail price, $7.49. The reformulation was blocked. Board minutes document the decision. The product is unchanged.
He didn't make the chemistry, he just refused to interrupt it.
Which is a decision. Countermeasure.
Rosemary. Salvia rosmarinus, 2 active phenolic compounds, rosmarinic acid and carnosic acid, 3 mechanisms. Rosmarinic acid scavenges superoxide radicals at the mitochondrial membrane. Carnosic acid activates the NRF2 pathway, rebuilding glutathione, peroxidase, and superoxide dismutase inside the mitochondrial matrix. And carnosic acid inhibits CYP2E1, slowing new glycidamide adduct formation at the source. Intercept the radical, rebuild the defense, slow the conversion simultaneously.
Cellular energy output degrades incrementally. The working adult who cannot explain their exhaustion after adequate sleep is experiencing a measurable bioenergetic deficit. They are producing less ATP than their baseline should generate. They will be told to sleep more, stress less, exercise harder. Nobody will evaluate what they ate for lunch.
And the rosemary extract, is that something you can actually get, or is this pharmaceutical grade?
Available in standardized supplement form. The specification, carnosic acid percentage must appear on the certificate of analysis. Steam distillation captures volatile aromatics, not phenolic diterpenes. Only supercritical CO₂ or standardized ethanol extraction preserves carnosic acid at therapeutic concentration, minimum 5%. If that is not on the label, the mechanism is unconfirmed.
Viper, the source.
Drôme, Provençale, southeast France, Buis-les-Baronnies. I'm already moving. 6 hours later, Buis-les-Baronnies, Drôme, Provençale, France. 4 centuries of phenolic extraction in this valley. They've known the difference between an aromatic and a therapeutic since before anyone had the language to explain why it mattered. The steam distillation units are here, essential oil production. Over there, the CO₂ extraction system. That's where carnosic acid survives. I'm looking at a batch certificate. Carnosic acid, 6.2%. Extraction method, supercritical CO₂, subcritical pressure, 40 degrees Celsius sealing. This is the specification. This is what has to be on the label.
Confirmed. That concentration delivers rosmarinic acid and carnosic acid at the therapeutic range used in the 2014 Romo-Vaquero study. CYP2E1 inhibition pathway is active at this dose.
And the steam distilled product, the one in most retail rosemary supplements.
Negligible carnosic acid content. A consumer purchasing a steam distilled rosemary supplement has an aromatic product, not a mitochondrial intervention.
N3. Confirm CYP2E1 inhibition threshold for carnosic acid at 500 mg standardized extract.
Confirmed. 500 milligrams of standardized rosemary extract at minimum 5% carnosic acid produces measurable CYP2E1 inhibition within 90 minutes of ingestion. Sustained inhibition across a 6-hour window at twice daily dosing. Acrylamide to glycidamide conversion rate reduced by approximately 40% at this dose in hepatic tissue models.
Understood. Viper secure the batch certification and move to extraction.
Already done. Moving.
The operative enters with a fat-containing meal. 2 molecules, different speeds, different routes. Rosmarinic acid water-soluble clears the intestinal brush border within 45 minutes. Passive diffusion, portal vein. Plasma concentration peaks at 90 minutes, already moving toward the mitochondrial membrane before carnosic acid has finished packing its bags. Carnosic acid lipophilic takes the slower route. Boards a chylomicron in the enterocyte. Lymphatic channels, thoracic duct, systemic circulation. Peak plasma at 3 to 4 hours. Destination, inner mitochondrial membrane. Its lipophilic structure means it belongs there. In hepatic tissue, first major site of acrylamide metabolism, carnosic acid reaches CYP2E1 in the endoplasmic reticulum. It binds. The enzyme slows. Glycidamide formation rate drops. The adduct production line interrupted at its source. Simultaneously, carnosic acid releases NRF2 from KEAP1 sequestration. NRF2 moves to the nucleus. Antioxidant response element activates. Glutathione peroxidase. Superoxide dismutase. Both synthesized inside the mitochondrial matrix within 12 hours of the first dose. And at the membrane, rosmarinic acid is already there, scavenging superoxide radicals leaking from compromised complex 1 subunits. The radicals that were oxidizing cardiolipin, propagating damage outward, they meet rosmarinic acid instead. The chain reaction stops. The complex 1 subunits already adducted do not repair overnight. but the rate of new adduct formation slows. The radical load decreases. Cellular ATP output begins to recover measurably within days of consistent intake. The exhaustion that did not respond to sleep, to stress management, to another cup of coffee, it had a molecular address. The operative found it. The turbine is running again.
Extraction certificate secured. Batch specification on record. Sourcing standard confirmed.
And the formulator's product?
Still on 6,200 shelves. The board minutes are dated 2018. The reformulation method that would have reduced acrylamide formation by 90% costs 4 cents a unit. It was never implemented.
The metabolic cost is not 4 cents. It is measured in mitochondria, in ATP, in decades of cellular energy production, running at diminished capacity. The individual who spends those decades in low-grade, inexplicable exhaustion will never receive a diagnosis that traces back to a board meeting in 2018.
Which is precisely the point.
Transmission intercept, fragmentary, authenticated as formulator origin.
Raryl hydrocarbon receptor activation confirmed at nanomolar concentrations. Immune tolerance cascade initiated within 96 hours. The compound is heat stable, survives standard pasteurization, scaling through the ready-to-drink protein beverage category. The demographic self-selects. They are already immunocompromised and looking for functional nutrition. Supply chain integration is complete.
Signal lost.
Log it. Protein beverage category. R pathway. He's already positioned.
He always is. The EPIC data goes into the mission file. 521,000 subjects. 30 years. The acrylamide fatigue correlation will eventually be published by someone. It should have been published a decade ago.
The timeline of publication is not our jurisdiction. The timeline of the next operation is aryl hydrocarbon receptor, protein beverage category, immunocompromised demographic. Mission 013 is authorized.
He is using their illness as a distribution mechanism.
He always has. That's why we don't stop.
Everything you're about to hear has been published in peer-reviewed research. I'm going to give you the mechanism, the study, and the exact dose in plain language. Acrylamide forms in starchy foods cooked above 120 degrees Celsius standard oven temperature. Your liver converts it to glycidamide, which attaches permanently to proteins inside mitochondrial complex 1, the first enzyme in your cell's energy production chain. Compromised complex 1 means less ATP. That is measurable fatigue, with a specific molecular cause. Rosemary extract counters this on 3 tracks. Carnosic acid slows the liver enzyme that makes glycidamide, activates your cell's own antioxidant production inside the mitochondria, and rosmarinic acid scavenges the superoxide radicals that damaged complex 1 generates. Romo Vaquero and colleagues 2014, Food and Chemical Toxicology, Volume 73. Rosemary extract supplementation significantly reduced acrylamide-induced oxidative stress, restored mitochondrial membrane potential, and suppressed CYP2E1, mediated glycidamide formation in human hepatic cell models. Supporting Pedersen and colleagues 2009, Mutagenesis, Volume 24, Issue 6 inches 475, non-smoking adults. Dietary antioxidant intake was inversely associated with acrylamide hemoglobin adduct levels. The mechanism holds in human tissue. 500 milligrams of standardized rosemary extract daily, minimum 5% carnosic acid, stated on the certificate of analysis. This is not present in steam-distilled rosemary essential oil and not present in most retail rosemary supplements because steam distillation captures aromatic volatiles, not therapeutic diterpenes. Look for supercritical CO₂ extraction or standardized ethanol extraction. If the extraction method is not stated and the carnosic acid percentage is not stated, the dose is unconfirmed. The label is not required to tell you this. So I am NBIA, Truth and Transparency in Human Health.